have reached the end of the replication cycle. Today we're going to do the last step, which is assembly. We're going to build some viruses. So we've made proteins, we've made mRNAs, we've made genomes, and today we're going to put it together, make some viruses. When you look at a virus particle, you should be able to tell how it's formed. Because remember, there aren't that many different kinds of virus particles. There are icosahedral particles that we have talked about. There are envelope particles. And there are nucleocapsids formed by nucleic acid and protein. So you should be able to look at the kind of particle and know exactly how it's formed. And that's what we're going to go through today, the way you build an icosahedral particle, the way you build an envelope particle, and the way you make sure that the nucleic acid is inside of it. All viruses have to go through a set of assembly reactions, which we're going to talk about. You make individual structural units. So if we're talking about an icosahedral virus, you make the individual units. They assemble by interactions, and you remember the principles of icosahedral interactions we talked about. Here they will come to fruition. You have to package the genome. Some viruses get an envelope, not all of them, of course, so this is in blue. The colors yellow here means every virus has to do this. The blue colors mean not all viruses. And they have to be released from the cell. Now, this also doesn't happen for all viruses. Some viruses stay inside the cell, and they have to mature. And the order may be reversed. Sometimes the viruses mature in the cells. Sometimes they mature outside of the cell. So we're going to talk about these reactions today, how they occur, and some of the common principles that are involved. And just like every other part of the replication cycle, assembly depends on the cell. It's not just happening spontaneously. It's very much like protein synthesis, nucleic acid synthesis, everything else. It needs things from the cell. And these include chaperones. A chaperone is a protein that helps another protein to fold properly. And cells have many different sorts of chaperones, and many viral assembly proteins utilize them. And in addition, some viruses encode their own chaperones. The assembly requires transport systems. The components have to get around the cell to the sites of assembly. And remember, movement within a cell is not by diffusion, never. It happens by active transport on motor pathways. Secretory pathway is important for the transport of glycoproteins from the endoplasmic reticulum to the plasma membrane, where they're going to become part of envelope viruses. And you also need nuclear import and export machinery. Right? mRNAs, of course, come out of the nucleus. Proteins can go back in. Sometimes virus particles are assembled in the nucleus, and then they have to go back out again. These are just some of the cellular systems that are involved. At every step, the cell participates. A key principle when we talk about assembly is concentration. The more concentrated the assembly subunits of the viruses are, the faster the reactions will occur. I like to say nothing happens fast in dilute solutions. So virus replication has focused on trying to find ways to concentrate materials in the cell. And one of the ways is that assembly often occurs in what we call factories or inclusion bodies. So these are discrete foci, either in the cytoplasm or in the nucleus, where viruses are assembled. And many years ago, before people understood anything about viruses and their replication, people would take cells from a sick person and look at them under a microscope, and they'd see these inclusions. And a lot of them were given names according to the pathologist that was looking through the microscope. So here on the bottom right is a neuron infected with rabies virus. And here's the nucleus here. It's looking rather sad. And here's the cytoplasm around it. You can see very faintly these darker staining areas. These are inclusion bodies that are very characteristic of rabies virus-infected cell. And they're called Guarnieri bodies. No, not Guarnieri, Negri bodies, sorry. I got the wrong pathologist. Negri was a pathologist who found these, and he called them Negri bodies. Guarnieri was another pathologist. They're not all Italian, by the way, just happens to be the two that I'm quoting, for different viruses as well. So that's a way to concentrate proteins. Another way is to put the proteins on a membrane, and we talked about this for RNA synthesis. A lot of viruses carry out RNA synthesis on the surfaces of membranes, and they often induce these membranes in the host cell. Those membranes, of course, are derived from cellular membranes. Viruses don't make their own membranes. And by making RNA synthesis on the surface, it happens more efficiently, and then you assemble the particles on the surface as well. So for poliovirus, we've talked about the replication of viral RNA on these double membrane vesicles that are shown in the lower left of a virus-infected cell. You can also see the small dark dots. These are virus particles being formed in association with those membranes. So these are just two ways that viruses concentrate things so that reactions go faster. Now, viral proteins need to get to the right places in the cell, whether it be for concentration or for assembly purposes. And to do so, they have addresses in them. And this is nothing unusual. Cellular proteins have addresses as well. And there are all sorts of addresses shown here. They're membrane targeting signals, like signal sequences. And as we'll see later, you can put a fatty acid onto a protein, and that will direct it to a membrane if the protein happens not to have a signal sequence. There are what we call membrane retention signals, signals that keep the protein in a membrane, nuclear localization signals, and nuclear export signals. And these are often pretty short amino acid sequences. For example, on the bottom is shown two different kinds of nuclear localization sequences. Here's a simple one, just consisting of a number of basic hydrophobic amino acids. This one's from SV40, from the T antigen, large T. And this, of course, gets large T into the nucleus. This NLS interacts with the import machinery. There are more complicated ones as well, but the principle is the same. The address gets the protein where it needs to be. So here's an example of import into the nucleus of a number of different viruses. We have influenza virus. The nucleoprotein is being translated in the cytoplasm. This needs to go into the nucleus because it needs to assemble there with the viral RNA that's being made in the nucleus. And that protein, the NP, has a nuclear localization signal on it. And so that brings it through the nuclear pore. Poliovirus capsids are assembled in the nucleus. These are DNA viruses like SV40. The subunits are made in the cytoplasm. They're shipped into the nucleus for assembly. They have NLSs on them, as is the adenovirus capsid subunit. Here's a hexon, which has been imported in. It has a nuclear localization signal. So again, for those viruses that need to assemble in the nucleus, mainly the DNA viruses, and in this case influenza virus, they have NLSs to direct them there. Many viruses bud from the host cell. They are enveloped. In this process we'll talk about in some detail in a moment. And these viruses, if you remember, have glycoproteins in their membranes. And those glycoproteins originate in the endoplasmic reticulum. They are made by ribosomes attached to the ER. They're translated into the lumen of the ER. And then they are transported to the plasma membrane by the vesicular transport system of the host cell. Vesicles break off from the ER and they fuse with the Golgi and move through the Golgi, and then these vesicles move towards the cell surface and fuse there, and then the viral glycoproteins are delivered on the cell membrane. And again, all this movement occurs on microtubules. And we don't always draw it in these pictures, but it's there, it's always happening. Sometimes it gets too crowded if we draw all the microtubules present, so we leave them out, but always movement of things in the cell is always occurring on microtubules. Now, let's talk first about the assembly of a virus particle. A common theme in many viruses is that the final virus is made up of sub-assemblies. You make proteins, they assemble into something that is not the final virus particle, and those sub-assemblies then make the final virus particle. And I want to show you three different strategies for making these sub-assemblies. The first is very simple. You make individual protein molecules and they combine. So we're using as an example here a subunit of the SV40 icosahedral capsid, a pentamer. And this is composed of 3 different proteins, 5 copies of VP1, and a copy of VP2, VP3. So the VP1 and the VP2/3 proteins are translated in the cytoplasm. They are imported into the nucleus and assembled into pentamers. This assembly actually occurs in the cytoplasm, and the pentamer is then imported into the nucleus. So the pentamer is a sub-assembly. It's obviously not the virus capsid. And pentamers will then assemble to form the capsid. Here's another example, adenovirus, the proteins that form the penton. These are the fibers at the 5-fold axes of symmetry that make the virus look like a satellite. They're assembled from individual proteins. Protein 4 forms the fiber. It's translated. It trimerizes. And then protein 3 is a subunit of the penton base, and 5 of those make the base. So these are actually sub-assemblies in themselves, and they form a second sub-assembly, the fiber joined to the penton base. So these are 2 examples of a simple way to make sub-assemblies. You make the individual components, and they come together. Now, the information for this coming together is encoded in the sequence of the protein. These proteins know how to fold properly and assemble with one another. That's how they have evolved. There's no magic involved. Sometimes we can make sub-assemblies from a polyprotein precursor. And this happens with viruses like poliovirus that make a long polyprotein and then cleave it up. So here, for example, we're translating a precursor to the structural unit of the capsid. And this consists of VP1, 2, 3, and 4. So the protein precursor is translated. As it's being translated, it actually begins to fold into its proper conformation. Here you have folded precursor. It's called P1, precursor number 1. You can see the VP1s, 2, and 3 have all folded up properly. And then finally, the protease, a viral protease, will cleave the loops linking these together. And that gives us a structural unit. And again, this is a subassembly of which there are many that form the viral capsid. So the folding of this is spontaneous, and then their assembly into a capsid is spontaneous as well. Now, sometimes we need chaperones to push this process along. And these can be viral chaperones or cellular chaperones. And these are simply proteins that help another protein to fold properly so they don't misfold. And here's an example of the assembly of a hexon trimer of adenovirus. So this is made up of 3 copies of this protein number 2. And the chaperone is called the L400 kilodalton protein. This is simply a virus protein that helps protein 2 trimerize and fold properly. There are many examples of this throughout virus assembly. So while there are plenty of examples of structural proteins that fold perfectly without a chaperone, there are also ones that need chaperones. We're not sure why some do and others don't, but again, they can be viral or cellular chaperones. So here's an example of the whole assembly process for poliovirus, one of the viruses we've just talked about. The capsids, of course, are icosahedral. You start with infection, which we have talked about before, the deposition of the RNA, the plus-stranded RNA in the cytoplasm. It's translated into a long protein, a polyprotein, which is then cleaved by proteases. And the P1, the precursor number 1, is relevant to us today because we're talking about assembly. And as I've just shown you, the P1 precursor folds spontaneously and then is cleaved. And here is that same pathway which I just showed you, except we know that it involves a chaperone. And I'm showing you a cellular chaperone here, HSC70, participating in the proper folding of this protomer, and a second chaperone here participating in the proper cleavage. So that subunit is just one of many subunits, of course, in the capsid. 5 of these come together to form pentamers, and then the pentamers assemble into a virus particle. And we think the genomic RNA associates with one of these pentamers, and that brings it into the viral capsid. All right, so it's a straightforward process involving a simple precursor that's cleaved to form a protomer, and then many of these assemble to form the capsids. So this is what we call an assembly line procedure. I've shown you this just for polio. You make a protein, it's cleaved, it folds properly into a bigger subunit, a pentamer, and those assemble into an even larger capsid. This is an assembly line because we're adding something along the way. Here's an even more extensive example of an assembly line process. This is for a bacteriophage, one of the tailed bacteriophages, very unique. They have icosahedral heads, helical necks or tails, and these little fibers here. And all of these are made as separate proteins. For example, the tail begins with a number of different proteins. Each number here is just the name of a different protein. They assemble first to form a base plate. Then the base plate assembles with a tube, and then that's wrapped with additional tubes to form the final tail. This is an assembly line process. Same thing with the head. The head is formed by the assembly of a number of different proteins to form the icosahedron, which eventually matures, and then it's attached to the tail. And the tail fibers go through a series of intermediates as well. And the advantage of this is that if you make a mistake, you can stop the process. If you make a defective base plate, it won't get incorporated into the rest of the procedure, and you won't make defective viruses with defective base plates. If you make a broken head, for example, it won't be placed on. So the assembly line process assures quality control in a way. You can't go on unless what you've made so far is good. And this is, in theory, what happens with automobile manufacturing, right? But of course, since humans are involved, you can make mistakes, and it's not like a self-assembling virus where the mistakes can be corrected. Here's an interesting example of an assembly line process where a scaffold is involved. Now, you know, when you build a building, you put a scaffold on the outside so you can walk up and attach things to the side of the building. And it's the same idea here, except the scaffold's on the inside. So this is the building of a herpes virus. And we make all the components of these capsids, which are quite large, in the cytoplasm. And you can see all the proteins here. They're imported into the nucleus, which is where the virus is going to be assembled. And then the subunits assemble into what's called a procapsid. Procapsid is the precursor to the icosahedral virion of herpes viruses. And inside this procapsid is a scaffold. You can see made up of a number of different proteins. You can trace them all to the key on the left here. It's an internal scaffold. Apparently this capsid is so big that requires a scaffold to keep it in shape, to build around it until it's done. When the capsid is done, there's actually a protease which is part of these scaffolding proteins. It's called VP24. It's shown over here. It's released from the scaffold and then digests the rest of the scaffold away. And presumably these amino acids that result from digestion leave the capsid through the portal. Remember, the herpes viruses are unique in that one of the five-fold axes has a hole called the portal in which the DNA goes in, as shown here. We're putting genomic DNA in here now. Presumably the scaffolding digestion comes out, and then when these viruses infect cells, the DNA comes out of the portal as well. So these establish an assembly line process as well, but the scaffold is needed. It's used, digested away, and then you have a DNA put in its place. So really, really amazing evolution of how things happen. So those are all examples of assembly line assemblies, or what we call sequential assembly. You make parts, you put them together to build a bigger and bigger structure. There's also another conceptually different form of assembly, which is called concerted assembly, because it happens all at once. It doesn't happen in stages, although some of the parts are made separately. But the whole virus particle is built in one step, all right? So the particles assemble only in association with the viral genome. That's the definition of concerted assembly. Whereas before, we would build the particle and put the genome into it. And this is exemplified here by influenza virus particles, which form by budding. And we'll talk more about this in some detail, but as I mentioned before, the nucleic acids, the RNA segments, are replicated in the nucleus. They join up with a variety of proteins, the nucleoprotein and the M protein, and they have export sequences that bring them out into the cytoplasm and target them to the plasma membrane. We'll talk about how that works today as well. The glycoproteins are made through the secretory pathway. They're also put in the plasma membrane, the hemagglutinin, the neuraminidase, the ion channel. And then this entire assembly buds out to form a virus particle. So the genome and the membrane proteins come together and it buds out, and that's the particle being made. That's why we can call it concerted, because the particle is formed in basically one step, this budding step. And we'll look again at the details of how this happens. But now we can distinguish two kinds of assembly, a sequential assembly line type of process in this concerted assembly where it happens all at once. That hemagglutinin and the neuraminidase, those two glycoproteins of the virus, are typical transmembrane glycoproteins. We've talked a little bit about them before in terms of their structure. Here's the virion of influenza virus. And you can see indicated the hemagglutinin, this purple club-shaped molecule, the neuraminidase in yellow. And on the right is the three-dimensional structure of the HA. We talked about how this is a trimer and how it facilitates not only binding to receptors, but fusion of the virus in the cell membrane as the pH drops. Really remarkable pH-catalyzed fusion. And here's just a description or a line drawing of the hemagglutinin. It's a transmembrane protein. So there's a short part of the protein that's in the interior of the virus particle, and this is very important for directing the structural components of the nucleic acid to where these membrane glycoproteins are inserted. And then there's an extracellular, extraviral part shown in red here, because it's modified in many ways with glycosylation. There are disulfide bonds to maintain the tertiary structure. There's a signal sequence at the N-terminus which directs it to the secretory pathway. And you can see here this green portion is the fusion peptide. So as this protein is initially made, the fusion peptide is buried. It's not even cleaved. And eventually this will be cleaved at this arrowhead right there before the end, and liberating the N-terminus of the fusion peptide so that at low pH you can insert it to membranes. So this is produced in the endoplasmic reticulum and then moves to the surface of the cell through the secretory pathway, which is shown here. So here we're starting out in the ER. The rough ER, the ribosomes are translating the hemagglutinin glycoprotein into the lumen of the ER, and the protein is folding as it's moving in. And then the vesicles break off from the ER and move that protein through the Golgi stacks and eventually to the plasma membrane. And of course, as the protein moves through the Golgi, a number of modifications occur, like glycosylation, oligomerization, glycosylation, trimming of sugars, addition of various sugars as it moves throughout. For some viruses, depending on the cleavage site of the HA, the cleavage may also occur in the trans-Golgi network. We talked earlier, and we'll talk in more detail later on, how the HA has to be cleaved in order for fusion to occur. And that cleavage, the enzyme that does that, can be either restricted to the respiratory tract, as for most human strains of the virus, in which case the cleavage occurs extracellularly as the virus particles are released. But for some highly pathogenic avian strains, the cleavage occurs ubiquitously by proteases that are present everywhere, and that happens in the trans-Golgi here. And the relevance of that to pathogenesis will become clear later on. So this is very typical of viral glycoproteins. They're produced in the secretory pathway. They're modified as needed. They're oligomerized as needed. And eventually, they may make it to the plasma membrane. I say may because some viruses actually mature at different parts of this secretory pathway, as we will see. Not all viruses bud from the plasma membrane. Some bud from the Golgi, in fact. Some bud from the ER. Some even bud from the nuclear membrane. So this is just an example of what occurs for some viruses. First question is, sub-assemblies are involved in which of the following types of virus particle production? Concerted assembly, sequential assembly, assembly lines, chaperone-assisted assembly, and all of the above. So 88% of you said all of the above, which is the correct answer. All of these processes involve sub-assemblies, concerted assembly, even though the virus particle and the genome come together at once. The genome assembly with its proteins happens in the nucleus separately in the example of influenza I gave to you. It also happens for sequential assembly lines and chaperone-assisted assembly. The point is that sub-assemblies are really involved in most virus assembly procedures. Those are two general ways to build particles: sequential processes and concerted processes, which largely involve budding. An important process in virus assembly is getting the nucleic acid into the particle, and only getting viral nucleic acid in. You don't want cellular nucleic acid in there, right? Because those particles will not be infectious. And often particles can't have both virus and cellular nucleic acids in them, so if you put cellular in, you're not going to have an infectious particle. And this occurs for both DNA and RNA molecules, because in the cell there's DNA and RNA, and you want to not package those, you want to package the virus nucleic acid. And the solution apparently is what we call packaging signals, and these have been identified in all kinds of DNA and RNA viruses, largely initially by mutation, and then later on by biochemical experiments. So here are two examples of packaging signals for two different DNA viruses. This is adenovirus on top and SV40 on the bottom. Remember, adenovirus genome is a long double-stranded DNA molecule. We're looking at the left end, which has one of the origins of replication, has the inverted terminal repeat. And you may remember there was a promoter not too far from the left end of the genome for the E1A, immediate early protein, the mRNA encoding it. And here are the packaging sequences, these blue arrows. And you can see there are a number of them, and they're scattered around. Some of them overlap. They also happen to overlap sequences here indicated as enhancers. These are long-distance sequences that influence the transcription of the E1A gene. So these packaging sequences are rather complicated, and the way they're identified is by introducing mutations into this region and ask, does this mutation block the incorporation of DNA into the capsid? And that's what is defined here. These are all the areas where you can introduce mutations that prevent packaging. So the way they work, they interact with a viral protein, which then pulls the genome into the capsid. In particular, for adenovirus, these sequences interact with the 4A2 protein, which then presumably gets the DNA into the building virus particle. On the bottom is the SV40 packaging signal. And remember, the SV40 genome is circular. Here's the origin of replication, right, which is the site where DNA synthesis proceeds in two directions. It's bidirectional. There's an early transcription unit to the right of the origin here. And here are some of the transcription factor binding sites. These are SP1 binding sites. And look, they overlap the packaging signal. So this sequence here, indicated by the arrows, if you mutate anything within there, you prevent DNA from being incorporated into the capsid. All right? So again, the idea is that these DNA sequences are able to bind viral proteins and get the DNA into the capsid. So these sequences are not present in the genome. And so genomic DNA is not packaged. Although if you took these sequences and put them into a piece of genomic DNA, you could get that package. And in fact, that's one of the kinds of experiments that's done to further confirm that these are packaging sequences. You can take a bacterial plasmid and put either SV40 or adenovirus packaging sequences into it and show that it will get incorporated into the virus particle. All right, so you can confer packaging to another DNA. And in fact, that is the basis for gene therapy vector building, which we'll talk about at the end of this course. People make viruses of all sorts to treat diseases, to cure cancer, to immunize, and you use the packaging signals to get your DNA into the virus particle. It's a really interesting field. The herpes virus genome is also packaged by these DNA packaging signals. But in this case, it's a little more complicated. And I want to show you this because it's very interesting and involves not just the packaging signals, but a head full of DNA. Now remember, the genome of herpes virus which is a double-stranded linear DNA in the virus. It comes into the cell, it circularizes, and replicates by rolling circle. So you make these very long concatemers, which are simply long DNA molecules with multiple genome equivalents in them. All right, so there's one genome length and another, and so forth. And the genome length, which I call a head full, is one of the determinants of packaging. So what happens is after replication has occurred for a while, you have very long molecules. You've built a few capsids. Remember, we just showed how that happens in the nucleus with a scaffold in the inside. You're now ready to put DNA in. There's a packaging signal at these sites called A in the genome. A here, and A here. And at the left end, this is what it looks like. It has direct repeats 1 and 2, and it has packaging signals PAC1 and PAC2. So PAC1 and 2 are needed for recognition of DNA by the portal. So here is a brand new virus particle. It's empty. It's got a portal on it. The portal, together with some other viral proteins, which are cut off at the top here, but all of that recognizes the packaging signal, PAC1 in particular, on the DNA. And then the DNA starts to get pumped into the capsid. There's probably a motor associated with this portal that basically pulls the DNA in. This is an energy-dependent process. And when we get a head full of DNA, which is signified by the presence of another set of these packaging signals right here, then there's a nuclease associated with this portal assembly that cuts the genome, and now we have a packaged genome. Now, you need to have two signals, the packaging signal and a head full. So somehow the machinery recognizes that we've put an entire genome equivalent into the capsid, and then it cleaves. Because remember, this packaging sequence is present at these other sites in the genome, but you don't want to cut there because that would make you a shorter than length genome. So in particular, that internal one, right? So two determinants. The packaging signals are recognized by the portal. That starts the import. And then when the capsid is full, essentially, then the cleavage can occur to give you a head full. So sometimes signals beyond the packaging signals are needed. And in this case, it's having a head full. And a head full is actually the modus for many different DNA-containing viruses. Now, RNA viruses also need to have packaging signals because you don't want to package various cellular RNAs like mRNAs. And here's an example of a packaging sequence of a retrovirus, HIV-1. At the bottom is a diagram of the left end of the viral RNA. This is actually part of the LTR and then the start site, the AUG. And the packaging signal is shown by a psi, the Greek letter psi, PSI, P for packaging. And this has been identified by mutagenesis as being important for packaging. You also need other sequences in the genome as well. In other words, if you just take this packaging signal and put it into another RNA, it won't get packaged. You need some other things as well. But those other things are important. They're known. And that's why we can actually make vectors based on HIV to do gene therapy. And again, we will talk about that later. This is the RNA sequence in this psi packaging region. You can see it forms a variety of stem loop structures. And in fact, this particular sequence here in stem loop 1, SL1, forms what is called, so romantically, a kissing loop complex. So two of these actually base pair. And remember, in each retrovirus particle, there are two copies of the RNA genome. And this is how you get two, by base pairing in this packaging sequence between this loop sequence that's shown in red. And the formation of that kissing loop complex then allows the viral nucleocapsid protein to bind, and that will incorporate it into the growing particle. We'll see that in more detail later, but here is the precursor of the nucleocapsid NC. It's called the GAG protein. The GAG protein is the first one translated from that AUG, and it includes the matrix protein, which will line the membrane, as you'll see, the capsid, which actually forms the shell of the virus particle, and the nucleocapsid. And the nucleocapsid protein will coat the RNA in the particle, and that is what interacts with this secondary structure, this kissing loop complex. And it does so by virtue of cysteine histidine boxes. They're called CH boxes. These are very good at interacting with nucleic acid. And you can see this area is important for RNA binding and packaging. So again, you form a complex between two RNAs, and that complex is then bound by the nucleocapsid, which brings it into the particle. And how that happens, you'll see in a bit. It's very interesting. So that's an RNA virus with a single genome molecule. And picornaviruses, paramyxoviruses would be similar. But what about a virus with a segmented genome where you have 8 or 10 or more pieces? How do you make sure that you get all of them in the particle and not just 8 copies of segment number 1, right? Or 5 of one and 5 of another. You have to have 1 of each segment in order for that influenza virus particle to be infectious. So here's a diagram of influenza virus particle. This is an envelope virus, of course, hemagglutinin neuraminidase in the envelope, and inside is the ribonucleoprotein. That's the RNA wrapped up with a number of different viral proteins. And there are 8 pieces, and each of those needs to be present in the particle in order for the particle to be infectious. So that's a really interesting problem, how you make sure that every particle gets 8. So over the years, people have had 2 hypotheses on how this happens, how you package 8 and only 8 genome pieces into the particle. And the first was actually a random idea, that every virus just grabs 8, and some of them are going to be non-infectious because you don't have 1 of each. But it turns out that if you do a random mechanism, you get 1 infectious particle for every 400. If you only limit it to 8 per particle, this is actually within the known particle to PFU ratio of the virus. So investigators thought, well, this could work because we know there are lots of non-infectious particles in every preparation of the virus. So maybe those are ones that just didn't get the right assortment of RNAs. So that stuck for many years. But now there's much better evidence that there's actually a specific packaging sequence on each RNA segment that mediates their incorporation into the particle, and that's shown here. What was really the key observation is that a study was done a couple of years ago which showed if you grow influenza viruses, every particle, over 95% of the particles, always have 8 segments. They don't have fewer and they don't have more. So there's clearly a mechanism for getting 8 segments in. In addition, if you look at the virus particles by electron microscopy, the segments are always oriented perpendicular to how the tip of the virus as it budded out of the cells. So if you just look at the bottom EM here, you can see there are 8 RNAs in them, and they're all oriented parallel to one another. You can see them because you can see the cross sections. And on the top is a model of how this happens during budding. Here's the forming virus particle, and the viral glycoproteins are on the surface of the plasma membrane of the cell. And then these ribonucleoproteins are oriented perpendicular to the budding tip, so that the budding tip would be the edge of the virus as it's emerging from the cell. So there's some mechanism that gets the RNAs oriented this way into the particle. And so genetic analysis showed that every RNA segment of the virus has a sequence at either end that contains a packaging signal. So there are 8 segments shown here from largest to smallest, and the proteins that they encode are shown. And at either end in blue are the number of bases that constitute the packaging signal. And so if you fool around with these, if you take them out of any particular segment, it doesn't get incorporated into the virus particle. Okay? In particular, what was done not too long ago, the packaging signals of the HA and the NS gene were swapped, and it works on different segments. So you can get HA packaged with an NS signal and so forth. So the idea is, and this is still a work in progress, we don't know how it works exactly, that these signals interact either with each other and or with viral proteins to make sure that only 8 segments get in, only the right 8 segments get in, and that no more or no less. All right, so that's what we know about influenza virus. We actually know a lot more about other viruses in terms of how they package RNA viruses anyway, and probably that will turn out to apply to flu as well. So let me tell you about one interesting example of what we call selective packaging. And probably this is what happens for flu, we haven't worked it out yet. This is a bacteriophage, Phi6, which infects bacteria. It's a spherical virus made up of 2 icosahedral shells. It's an outer capsid and an inner capsid. And that should ring a bell because the rotaviruses, the reoviruses, have a similar structure. Those are viruses that infect mammalian cells, but this happens to be a bacteriophage. And this virus has 3 double-stranded RNA segments. They're called small, medium, and large. And packaging obeys a serial dependence. In other words, when these shells are formed, the first RNA that goes in is the small RNA. Medium and large don't go in first. Apparently, they don't have the right signals. Once the small RNA is in, then the mRNA can get in, but not the L. And only when the mRNA is in, then the L can get in. So SML is the order of packaging of these RNAs. And that's dependent on interaction of the packaging sequence with proteins that constitute the capsid. We think that a similar mechanism occurs for the rotaviruses, which have 10 segments of double-stranded RNA. It's a serial mechanism. One goes in first, and then that directs the incorporation of other RNAs as well. And this may very well turn out to be how influenza viruses package their genome as well. But the point here, of course, is that whether it's a DNA or an RNA virus, you need to have a specific way to distinguish the viral genome from that of the host cell, and it's done by these packaging signals that interact with viral proteins to direct the genome to assembly. So the next question is, packaging signals on viral blank interact with viral blank during virus assembly. So you can put in lipids, proteins, you can put proteins and sub-assemblies, genomes and proteins, proteases and membranes, or proteins and genomes. Packaging signals on viral genomes interact with viral proteins during assembly. 84% of you got that correct. So genomes, the packaging signal on the genome is interacting with the protein. Maybe you got confused by the last one. Let's talk a little bit about the formation of enveloped viruses now, how you acquire this membrane. And this is a process we call budding. I've used that term before, and now we're going to see what it actually means. It happens in 2 ways, the formation of these particles. You can first assemble internal structures and then acquire a membrane. So we showed that for influenza virus, genome RNP is formed, and then the whole particle is formed by budding. And that happens for most envelope viruses. Or it happens with the assembly of internal structures in retroviruses. So the particle isn't actually formed until the internal parts are made, and we'll see how that happens in a moment. Depending on the virus, different viral proteins can drive the budding process. And what I mean by driving is, if you just take a single viral protein and produce it in a cell, is that enough to get a particle? So for some viruses, for example, this class 3, the envelope proteins alone are enough to make a particle. So influenza virus and coronaviruses, you can take a gene encoding the HA protein and express that gene in cells, and you will make particles. They'll be empty, but you will make particles which are little membrane vesicles with the HA on the surface. And that's actually being used to make a vaccine against influenza virus. And you can do this in plants. You can express the HA gene of flu in plants. You will get particles made by budding. You can purify those, and boom, you have a vaccine. For other viruses, it's a little more complicated. For example, in some, it's the internal proteins that drive budding, either a matrix or a capsid protein. So the matrix protein is typically the protein that abuts the viral membrane. It's shown here as blue spheres. And for some viruses, you can produce just that protein and you will make particles. For other viruses, you need both the envelope glycoproteins and the capsid. So either one is not enough. The capsid's not enough, the glycoprotein is not enough. And then for some, you need all sorts of determinants, not just matrix proteins, but glycoproteins and genome as well. So there are different requirements for budding, but the point is that in the simplest case, you can actually get particles made with one or very few viral proteins. So let's review again how this is happening with influenza virus. We are, again, making our genomes in the nucleus of the host cell. Of course, mRNAs have already been made, and these come out into the cytoplasm, and they are used to synthesize viral proteins, among which are the glycoproteins that get shuttled to the plasma membrane by the secretory transport system. We talked about that already. So these go up into the plasma membrane, the hemagglutinin, the neuraminidase, the ion channel. The viral RNAs that are destined for new particles have to associate with viral proteins. in particular the nucleoproteins that coat the RNA, and then the M protein, which is shown as a blue rectangle here. And these are exported out of the nucleus. They have nuclear export signals. They don't just diffuse, of course. They're moved on motor pathways, as we'll see in a moment. But the point is that they reach the plasma membrane and associate very tightly with it, and that eventually causes the particle to form by budding. And then the membrane pinches off at the base, and you have a free moving virus particle. So that's the process of budding, acquiring an envelope. We called this concerted assembly before because the virus is made when you have the budding process occurring. This movement, of course, of these nucleoproteins through the cytoplasm is by motor pathways. In particular, it's been found very recently that these nucleoproteins, which are again diagrammed as on this slide, going from the nucleus up to the plasma membrane, they move on the surfaces of endosomes. So the endocytic pathway we talked about in this course as originating by invagination of the plasma membrane, formation of endosomes, and movement towards the microtubule organizing center near the nucleus. But this pathway also goes in the other direction. And what are called recycling endosomes take bits of this internal endosome and bring it back up. So in fact, the viral nucleoprotein of influenza virus, after coming out of the nucleus, associates with this endocytic export pathway. And that's what actually brings these RNPs up to the plasma membrane. So again, it's not diffusion. It's always movement by motor proteins, and that's shown by these microtubules here. The motor proteins aren't shown, but they're certainly implied. What allows the M protein to associate with the plasma membrane? Let me go back and just emphasize what I'm talking about. The M protein, M1, is this blue rectangle, which gets imported into the nucleus, and it binds the viral RNP. That M protein is eventually going to form the inner layer of the virus just below the membrane. The question is, how does it find the plasma membrane? How does it attach to it? Well, the transport pathway brings this complex very close to the plasma membrane, and then the M protein has signals built into it to allow it to interface with the plasma membrane. And here are those signals shown on a linear diagram of the protein here. So the top in part A is influenza virus M1. So again, that's the blue rectangular protein that is bound to the RNA and is allowing it to associate with the plasma membrane. And you can see here on the protein are a number of domains indicated. Here's a nuclear localization signal. Right? So when this protein is made in the cytoplasm, it's got to get in the nucleus, and that's the signal that does that. There's a nuclear export signal, NES. Once it's in the nucleus and it's grabbed the RNA, it's got to go out again, so that's how it does that. And these two bracketed areas are very important. You can see they're both important for binding to the RNP. So if you delete these areas or make mutations in them, this M protein will no longer bind the RNP, will no longer bind the RNA. And this part on the left contains hydrophobic regions in orange, and that's important for binding the membrane. So these are the sequences of the proteins. They're highly hydrophobic, which allow the M to sit on the underside of the plasma membrane. It's not a signal sequence, but it's simply a hydrophobic patch of the protein. If you change those amino acids to non-hydrophobic amino acids, the protein will not associate with the membrane, and you will interfere with budding. So VSV, vesicular stomatitis virus, also forms by budding. These are those bullet-shaped virus particles in a very similar process. And again, it has an M protein that lines the interior of the membrane and which brings the viral RNA to the plasma membrane, just like that of influenza virus. And you can see it has sequences that are important for both membrane binding as well, the plasma membrane, a hydrophobic region, and then a positively charged region that allows the M protein to bind to the RNA. So it has two kinds of signals, one that binds the RNA and one that allows it to bind the plasma membrane. So this is what I meant earlier, that the virus proteins have addresses in them that allow them to go to the right place. In this case, the M protein has to go to the plasma membrane, so it has hydrophobic patches that allow it to do that. So that's influenza virus. Many of the envelope viruses that we've talked about, vesicular stomatitis virus, the paramyxoviruses, they all mature in a similar way. The retroviruses are slightly different. And I want to explain to you how that works. This will bring together a lot of the information I've been giving you about packaging as well. Now remember, the retrovirus genome is a plus-stranded RNA. It's made in the nucleus by transcription of the provirus. Remember, the provirus is integrated into our cellular DNA. The LTR on the left has a promoter that will result in the production of an mRNA. And that mRNA then gets exported to the cytoplasm. This is going to be a genomic RNA. And remember, at the end terminus, or at the 5' end of that RNA, there's a packaging signal which allows for dimerization of the RNA. So we have two RNAs here that are now brought together. And that dimerization allows binding of the virus nucleic capsid protein. So that's the little protein shown as a circle here. Now let's go and see where that's from. I've already showed you a picture of the NC protein, and I showed you the parts of the protein that bind to the RNA. And here it is, nucleic capsid is part of the GAG precursor. And there's NC. So as this protein is made, it remains a polyprotein. The individual domains fold, but it remains a polyprotein. So here, for example, we have a matrix capsid NC, P1, P6. These are these other domains here, which we'll talk about in a moment. See, one of those polyprotein units is going over here and binding viral RNA. So look at the M protein is MA. It's called MA for retroviruses. It's lined up here on top of the capsid. And once it binds RNA, then that protein, the matrix, has signals in it that target it to the plasma membrane. We'll talk about those in a moment. And it brings the RNA along with it. So here at the top left, you can see these are associating, these subunits of matrix, capsid, et cetera, are associating with the plasma membrane. And one or two of them are associated with the viral RNA. You also make a longer polyprotein that goes beyond the end of GAG. If you remember, the reverse transcriptase integrase coding region is downstream of GAG, and you only make that either by a translational suppression or a frame shift. You get a longer polyprotein. And one of those is shown here. We have matrix capsid, nucleocapsid, protease, reverse transcriptase, and integrase. And a few of those also get incorporated. But you can see they're incorporated by virtue of matrix, MA, binding to the plasma membrane. Okay, so as this patch begins to bud out, more and more of these proteins are recruited to coat the membrane. We've got two genomes in here. You don't often put many of the longer precursors. Remember, there are only 50 to 100 molecules of reverse transcriptase per virion. So in the whole circle that's going to form here, you're only going to have 50 to 100 of those. And then it buds out. Now what's formed here is a particle that has matrix and capsid. It's got two molecules of RNA. It's got reverse transcriptase, integrase, and protease. So protease is crucial here. That is the yellow sphere, because then once the virus is bud off the cell, the protease then cleaves all of these precursors. Remember, they're incorporated into the particle as a polyprotein with the individual domains folded properly, but after the particle is made, the protease comes in, cleaves all the proteins, and now the particle matures. And now you see it has an actual capsid. The matrix is lining the envelope in blue, and the RNA, the reverse transcriptase, integrase, et cetera, are all inside. So this is different from all the other envelope viruses. So these particles don't form on budding, they form after budding when the internal parts mature by the action of this protease. But the neat part here, again, is that the RNA is brought in by virtue of its binding to nucleocapsid. We showed the basis for that earlier, binding to the packaging sequence. And then all of these other proteins are brought in by virtue of them being part of a polyprotein. Remember, the bonds between them are not yet cleaved. That's how they all get into the growing particle. Turns out that if you just produce GAG protein, which is this, in a cell, you can make virus particles. And that's also the basis of viral gene therapy. You can make retroviruses with any gene in them. You just have to produce GAG. And then you take the packaging sequence of the retrovirus and put it on the gene that you want to be incorporated. You will now have a virus that can infect cells. It won't replicate because it has many viral genes, but it has a gene that you want to deliver. If you want to deliver a gene to correct a genetic disease, for example, you can package it in these particles. So we're going to talk about this at the end in gene therapy, but this is the basis of it. This basic biology that people figured out allowed us to make these vectors. I want to show you how the matrix figures out how to go to the plasma membrane. This is an incredibly important step, right? Because the matrix binding the plasma membrane brings all the other proteins there, brings the RNA there as well. So this interaction is crucial. How that happens is that there are some sequences at the N-terminus of matrix. Here is, again, the GAG precursor, matrix capsid, nucleocapsid. We talked about the RNA binding regions of nucleocapsid. Matrix has two patches, which are important for membrane binding. But more importantly, the N-terminus of the protein is myristoylated. This is a little lipid that is covalently linked to the protein. And that allows it to interact with the plasma membrane. Okay, if you take away myristate, you can modify the sequence of the protein so that it's not myristoylated. You will not get assembly because matrix doesn't know where to go. It's lost. It's just floating around in the cell. It will never reach the plasma membrane. So this myristoylation is crucial. So this is a lipid modification of a protein. It turns out these are very typical. This happens frequently in virus assembly. Proteins are modified with lipids. Here's myristate, by the way, in case you're interested. This is the myristate moiety here. As you can see, it's a long hydrocarbon, and it's usually attached to this kind of a signal in the protein. And this allows you to target a protein if it doesn't have a signal sequence. So the matrix is not made in the secretory pathway. It's made in the cytoplasm. So you can't, how do you target to a membrane? Well, this is one way. You put a myristate on it. Another way is to put hydrophobic patches as well, as we saw for flu. So these proteins, you make them in the cytoplasm, and then there are enzymes that attach the myristate. Myristate is one of many different lipid modifications that can be added to viral proteins to target them appropriately. Now, what about the budding process? You know, I've kind of just said magically these buds form, but it's no magic, right? It's biochemistry and cell biology, and we figured it out. Not me, but virologists, right? And it was because people many years ago made some mutants. Here's the GAG precursor of HIV and of a different retrovirus, a mouse retrovirus, Maloney leukemia virus. And people were mutating this thing called P6 here, or P12 for the other retrovirus. You know, these are just people in the lab saying, what does this P6 do? So let's mutate it. Well, when they mutated it, they got these funny budding viruses that never detached. That's the phenotype of these viruses. They remained attached to the cell surface by this stalk. All right, so you make a mutation in P6 or P12, and that's the phenotype. This happened in part at Columbia. I remember watching students reporting these results. I'm looking at these pictures of these. I said, what the hell is going on? Well, it turns out this revealed the whole machinery of budding. It's really interesting. Turns out that this arrest happens at a late stage. So the domains that were mutated were called late domains or L domains. And after they were discovered in these retroviruses, very specific sequences, you can see PSTAP, LYPXL, those are amino acid. That's the sequence which was changed. But you can find them in other viruses as well that bud from the cell. So this is a universal signal for budding. And what it turns out is that these late domains, again, the green are the late domains, they bind cell proteins whose function is to split membranes. I'm going to show you that in a moment. All right, so during mitosis, vesicle trafficking in the cell, vesicles have to split apart. And there's a whole machinery that does that. And these L domains interact with that machinery. And so that's shown here on this slide. You can see these L domains are associating with a variety of different proteins. They're all part of an ESCRT pathway, ESCRT. And what does that mean? That is the endosomal sorting complexes required for transport. This is a pathway originally identified in yeast by mutation, and it turns out to exist in mammalian cells as well. When cells have to divide, it's a complex of 30 or so proteins that help do that. And in particular, the ESCRT3 component helps to pinch off those membranes. It's called abscission. When cells divide, it's called abscission. The ESCRT pathway is involved. Within the cell, vesicle trafficking also involves the ESCRT pathway. The formation of exosomes. Exosomes are little vesicles that are shed at the plasma membrane. And their formation from a multivesicular body requires membrane splitting, and that's carried out by the ESCRT pathway. So guess what? It turns out that budding viruses use this pathway. I can't use the word use, but it's the quickest thing I can think of right now, right? They have evolved to take advantage of this ESCRT pathway to make a bud. This was not its intention. The ESCRT pathway was not intended to make viruses bud, but viruses have adapted. They have viral proteins that bind the ESCRT machinery, as I've just shown you, identified originally by mutation. And then that recruits the ESCRT3 component, which pinches off the bud. It's just remarkable. This happens in just about every budding virus that we know of. So they have proteins that interact with the ESCRT pathway, and they usurp it to make the virus particles bud, when normally these things are required for normal cellular processes. So this is a remarkable system. It explains how viruses bud out of the cell. All right. So again, normally, this is not the function of this pathway. And it's just a beautiful interaction of viral and cellular pathways. Now that happens at the plasma membrane, but as I mentioned earlier, budding can happen at almost any membrane in the cell. Viruses can bud from the nucleus. So here, herpes simplex virus can bud from the nucleus because it inserts its glycoproteins in the nuclear membrane. Some viruses bud from the ER. Here's a coronavirus. The glycoprotein's inserted into the ER, and then the virus buds from there. Some viruses bud from the Golgi as well. And we've talked a lot about viruses that bud from the plasma membrane. All right, so it can happen pretty much anywhere. So if you're ever asked the question, where do viruses bud? Pretty much wherever there is a nucleus. And let us do this last question before exploring that. Which statement about viral budding is incorrect? The envelope can be acquired before or simultaneous with assembly of internal components. The viral spike glycoprotein can drive budding. No host proteins are involved in the budding process. Lipids assist structural proteins to interact with the membrane. Budding can occur from the nucleus, ER, Golgi, or plasma membrane. So 81% of you said no host proteins are involved in the budding process, and that is absolutely right. That's not correct. I just told you about the ESCRT pathway, of course, so that should stick out. A few of you answered all the others, but they're all right. The envelope can be acquired before or simultaneous with assembly. Before, most envelope viruses simultaneous with assembly, the retroviruses. The spike glycoprotein can drive budding for sure. Lipids assist structural proteins to interact with the membrane. The N-terminus of matrix is myristylated, and that drives it to the plasma membrane. And finally, how could you answer that that's wrong? I just told you that the budding can happen at any of those membranes. I have to assume that you're just fooling around. That's fine. Well, it's a good way for you to remember. You'll never forget that now, right? So herpes virus buds from the nucleus. And if you think about it a little, how do you get out of the cell if you bud from the nucleus? Because you're going to lose your membrane. If you just bud from the nucleus, then fuse at the plasma membrane, you'd be a naked nucleocapsid. And we know these are envelope viruses. So the machinations this virus has to undergo are remarkable. So here we go. Herpes virus capsids, remember, assemble in the nucleus. We talked about how you make a scaffold that gets digested away and the DNA gets put in. And that capsid has to get enveloped. Buds out of the nucleus. So in this wonderful diagram, there are electron micrographs at every step. So here are the capsids in the nucleus. This is budding from the nuclear membrane. So now you have the endoplasmic reticulum, you have an envelope virus particle. So what is it going to do? It fuses with the ER, and now you have the capsid out in the cytoplasm. So you have gotten out of the nucleus, but you still need a membrane. So what happens is this particle then buds into the trans-Golgi network, acquires a membrane, and then it buds out of the trans-Golgi. So now we have two membranes. You can see, look at these particles in the EM, they have two membranes. Beautiful. And then that outer membrane finally will fuse with the plasma membrane. And now what comes out is a virus with an envelope in the capsid. So it's perfect. There's that picture of it. And that fusion is, of course, mediated by the fusion protein machinery of the cells. So this is what you need to do if you bud in an unusual place. And you can think about budding from the ER into the cytoplasm and so forth. One more really interesting mechanism of exit from cells. Now, vaccinia viruses acquire envelopes by budding from within various cellular compartments. They actually make a double membrane enveloped particle, which then fuses with the plasma membrane. So vaccinia viruses are made within the cytoplasm. They move these double membraned particles. I'm going to show you how they're generated. That's not the point here. They move to the surface on the microtubule network. They fuse with the plasma membrane, and they remain associated with the plasma membrane. They stimulate, one of the viral proteins shown in blue stimulates, activates a kinase, which then modifies the actin network below the plasma membrane. So you know that beneath the plasma membrane of many cells is a microfilament network made up of actin, right? So vaccinia rearranges this to kind of launch it to another cell. So the virus activates this Src kinase. This triggers actin polymerization, and you get these big projections from the cell, which are full of actin filaments, and that pushes this virus to the next cell. So if you take infected cells with these filaments and you add uninfected cells, these viruses get pushed into them. And you can see on the upper left is a fluorescence photograph of an infected cell. The green are the actin filaments, and at the tips are the yellow virus particles. So basically the virus induces the cell to propel it to another cell so it can spread more readily. And this is on the right is a stain of the actin filaments so that you can see how they form, projecting out of the cell, and there's the virus particle at the tip. It's just remarkable. I had to show you this because there's nothing like it. And that's how the virus spreads from cell to cell. So this is a virus that forms by budding intracellularly, loses that outer membrane, and then gets shoved into another cell. Okay, what about non-enveloped viruses? How do they get out of cells? Budding is easy. You know, you make a bud at the virus surface, or you fuse and get shuttled out by membrane fusion. That all makes sense. But what about a virus like polio, where you have a naked capsid? There's no envelope. How do you get out of the cell? Well, in many of these viruses, the cells die. Here's a progression of cytopathic effect in cells infected with polio. The cells are rounding up, they're breaking open, and the virus gets out. And that's because many viruses induce apoptosis, necroptosis, another form of programmed cell death that involves the immune system. They kill the cells, and this facilitates virus release. There are also a number of virus proteins that rupture membranes. We actually talked about one of those in adenovirus that ruptures the endosome when the virus comes in. But a number of viruses make proteins that poke holes in whatever compartment the virus is in, could poke holes in the nucleus or in the plasma membrane, and that lets the virus get out. And these are typically called viroporins, pore-forming virus proteins. And they've been identified in polyomaviruses, influenza viruses, and HIV. And finally, the other contribution to cells breaking, remember, we have to get a non-envelope particle out of a cell. The best way is, would seem anyway, would be to break the cell. You know, these viruses all inhibit protein synthesis. This really leads to loss of membrane integrity, and eventually the cells develop holes and the viruses can get out. So these are what we think happen to enable non-envelope viruses to get out of cells. But more recently, there have been more inventive pathways discovered, and I'm going to end up by showing these to you. And these involve non-lytic release of non-enveloped viruses. So when enveloped viruses bud from the cell, this can happen over a period of many hours. The cells have to remain alive so that you can make lots of particles. You don't want to lyse the cell immediately after you make a few particles. That would not make any sense. But lytic viruses, well, they break the cell open when you get a certain number of virus particles inside. But a number of instances have been discovered now where these non-enveloped viruses don't actually lyse the cell. And one of them is shown on the top here, and this is for poliovirus. Now, early in infection, the virus induces the formation of double membrane vesicles, which are typical of the process called autophagy. When cells are subjected to stress, they induce autophagy. And these double membrane vesicles form, they encompass the cell cytoplasm, and then they actually bring it to the surface and release it. It's a way of recycling cell contents. Late in infection, these autophagosomes contain virus particles. And they mature by losing one membrane, and then they fuse with the plasma membrane, remember, in the process of releasing cytoplasmic contents. But if there's virus particles inside, those will get released as well. And it's been shown if you inhibit autophagy in a polio-infected cell, you will inhibit virus release to a certain extent. So this is one non-lytic mechanism of virus release, in particular of a non-envelope virus. Just about a year ago, an even more surprising mechanism was discovered, and that's shown on the bottom for hepatitis A virus, which is related to polio. Now, you may know, having taken biology, that cells produce little particles called exosomes. And these are produced from multivesicular bodies. They can contain proteins, lipids, microRNAs. They basically form by invaginations of the cytoplasm. And then they move to the surface and are released in their own vesicles. So this multivesicular body would fuse with the plasma membrane and release exosomes. It turns out that hepatitis A virus in liver cells, which is the right cell for hep A, it's a liver pathogen, is actually produced from cells within exosomes. So they have membranes. They have membranes. This is a non-envelope virus, but in the liver cell, it acquires a membrane by being released in exosomes. This membrane doesn't have any viral glycoproteins in it. The virus doesn't encode any glycoproteins. It's completely resistant to antibodies. Antibodies usually bind viral proteins. So this is a brilliant strategy for evading the immune system. And it's since been shown to happen with poliovirus as well. So it's a non-lytic way that viruses without an envelope can get out of the cells in these exosomes. Really remarkable. So that brings us to the end of the infectious cycle. You now know how to make a virus. And next time, we're going to infect some hosts and see what happens.